Good morning. I think we'll go ahead and get started while people are sitting down. It's my pleasure to introduce Sara Hart this morning, one of our newest faculty members. For those who don't know Sara, she obtained her bachelor's in biochemistry, probably from Knox College, and then did her PhD in immunology at the University of Iowa, and worked with John Hardy on CD8 T cell memory. Then in 2003, Sara came to the University of Minnesota to do a postdoctoral fellowship with Steve Jameson. And while she was there, she was awarded an ACS postdoctoral fellowship. And then when she completed her postdoc, she stayed on with Steve as a research associate. And during that time, she was a Leukemia and Lymphoma Society special fellow. Then in 2014, am I getting all these dates right, Sara? Good. Sara became an assistant professor in our department. Sara now is developing a really interesting new line of research on malaria. She's also been very involved in a project some of you may have heard about in the Center for Immunology, the Dirty Mouse Project, and she is now the director, in fact, of the Dirty Mouse facility, also called the Normal Microbial Experience facility, affectionately called the Dirty Mouse facility. So Sara has two really different projects going on, and I'm not sure what she's going to talk about today. Maybe it looks like the dirty, not malaria. Okay, that'll be for another time. Sara, welcome. Okay.
Thanks, Chris, and thanks to everybody for being here this morning and giving me the opportunity to share some of my current research with you. So my focus for today is really on CD8 T cell responses. So I wanted to start with just this classic slide that a lot of people show, in which demonstrates the vigorous expansion that naive T cells go through when they see their specific antigen. This phase is followed by a contraction period and establishment of long-lived memory cells. And the goal of creating these memory cells is really that they are able to more robustly respond than naive T cells when they see antigen a second time, shown here. So to achieve this better response, the memory T cells need to obviously survive to see that antigen again. They need to be able to expand. They express effector molecules that limit the proliferation of the pathogen in question. And ultimately, they participate in the clearance of that pathogen. And this is typically what I focus on most prominently in my research, because I feel like really achieving sterilizing immunity encompasses the success or failure of all of these different characteristics of memory cells. So it turns out that memory T cell populations are quite complicated, and the heterogeneous nature of this pool is increasing all the time as the research develops. Initially, there were two populations defined called central memory and effector memory. But over time, the work of Dave Massapust from here at the University of Minnesota and others also identified resident memory T cells as really important contributors to immunity in tissues. And a few years ago, we defined another population that I'm going to talk more about today called the long-lived effector cells. And since we defined this population, originally I've been interested in thinking about what are the unique contributions of this cell type to overall immunity. I've tried to really limit my use of cell surface markers in my talk today, because I know that can be sort of distracting if you're not familiar with all of them. But I just included this slide to illustrate that we're really defining these different populations based on the surface molecules that they express and also their migration patterns. So for example, I am going to mention several times this molecule CD44, and it's highly expressed on all memory T cell populations, so that allows us to distinguish them from naive T cells. And other markers that I've listed here are able to allow us to differentiate between these other subsets so that, for example, if I'm interested in long-lived effector cells, I can use these markers to isolate those cells and further define their characteristics. And one way that we really think about the unique contributions of memory subsets is to use the technique of flow cytometric sorting. So here I'm just showing an example where we can stain the surface of the cell with some of these markers that were on the previous slide using fluorescent antibodies. And then using sorting technique, we can isolate those into separate tubes, for example, and then perform further analysis on different aspects of their functional and gene expression characteristics. And first, I just wanted to show this example of principal component analysis we did after we sorted these different populations, isolated RNA, and performed RNA-seq. And the first point I want to make is that you can see the long-lived effector cell population is clearly very unique from central memory and effector memory cells. And I'll come back to some of the other highlights from this data in a few slides. And also, I should say, we didn't perform analysis on resident memory T cells, but others have shown that they also display a very distinct gene signature. Another way that it's very useful to use this technique of isolation of these different populations is when we ask about the protective capacity of each population individually. So in this example I'm showing, we've used a transgenic T cell called OT1 that's specific for ovalbumin. And we can transfer a few of these cells into a mouse, then infect it with an ova-expressing bacteria or virus, of which we have many varieties. And then we simply wait to allow the different memory populations to develop in the animal. Then, after here 90 days, we can use flow cytometry to isolate the different populations and then transfer them into new mice at equal number so that we can directly compare their ability during the secondary infection. And also I want to say on this slide that a lot of my data is going to be using Listeria monocytogenes infection as a challenge model. And that's because it's very straightforward to determine the bacterial load after infecting with this organism. And also CD8 T cells are required for sterilizing immunity. So it's a good model in this context. So then when we perform this transfer and challenge the mice, 5 days later in this graph, I'm showing the colony-forming units of Listeria in the spleen. And you can see that if we don't transfer any memory cells, this is the level of infection, which is pretty high in these animals. If we transfer the effector memory subset, there is a reduction in the bacterial load. But if we transfer the long-lived effector cells, there's a much greater reduction. And we've done this experiment a number of times, also adding in the central memory subset as well, and always the long-lived effector cells perform more robustly. So in the next few slides, I want to talk about how the long-lived effector cells are able to accomplish this increased clearance of the bacteria. So memory T cells have two methods or modes of becoming activated. The first is what we typically think of, where they recognize specific antigen. And here this is shown by an infected cell, which processes and presents antigen in the context of MHC class 1. And then when the T cell comes along, if the T cell receptor is specific for this class 1 peptide combination, it will become activated and certain effector molecules will be produced. For example, interferon gamma, which is a very important pro-inflammatory cytokine, and granzyme B and perforin will be upregulated, which will allow the cell to be cytolytic. Now there's a second mechanism of memory T cell activation that occurs independently of antigen recognition, and that is through cytokine stimulation, particularly through these two cytokines together, IL-12 and IL-18. These are produced very early during an immune response in the first few hours, and will also induce memory T cells, but not naive, to produce the same effector molecules. So this is an important mechanism for T cells to respond very rapidly during an immune response before processing and presentation may have had time to occur. And this is, I can't remember what I said, often this is called a bystander response, if you've heard that term used. So if we expose the sorted long-lived effector cells, or the effector memory cells, to these two cytokines, so this is just different concentrations of cytokine, in a short-term in vitro assay, you can see that both types of memory cells make interferon gamma, but it's really the long-lived effector cells that more robustly produce the cytokine. And the same is true for the frequency of cells expressing granzyme B. Now, this characteristic of rapid interferon gamma production is also a characteristic of NK cells. So again, those innate cells respond very rapidly during the first few hours of infection. So we wanted to go back to our RNA-seq data to see if there was any shared characteristics in that data set with NK cells. And this, in fact, we did find a lot of NK cell-associated molecules. So this is just showing a heat map of the top differentially expressed genes between the long-lived effector cells, effector memory, and central memory. And I just pulled out a few of the top hits, which are NK-associated molecules, and we've confirmed a lot of these by protein expression. Additionally, other markers you might be familiar with, NK1.1 and NKG2D, which are definitely associated with NK cells, are also upregulated on long-lived effector cells. So all of these together are an association with this innate cell type and may be an indicator of their ability to rapidly respond during infection. Okay, next I wanted to comment on the location of long-lived effector cells in the spleen and if that might play a role in their ability to respond more quickly. And this is just showing, as if you remember, the red pulp and the white pulp of the spleen. And typically we think of T cells being in the white pulp, but actually the red pulp can be a first site of pathogen capture for bloodborne pathogens. So we have a technique where we can inject a fluorescently labeled antibody, in this case CD8, wait a few minutes, and then sacrifice the animal and determine if the cells are located in the blood vascular compartments of tissues, also this includes the red pulp, or if they are protected from antibody labeling, they will be in the white pulp. So when we use this technique to identify where these cells are located, you can see that effector memory cells, there are a portion of the cells that are located in the red pulp, and I should say this is just at the steady state before infection has been initiated. But the majority of them are located in the white pulp. However, the long-lived effector cells are very strongly in that red pulp compartment, which may make them positioned in the best way to then come in contact with infected cells, especially in the case of my example with Listeria. Okay, so lastly for this first section, what about long-lived effector cells in humans? So not all of our mouse markers nicely correspond to human immunology, but this marker has recently come to light. So CX3CR1 was highly expressed in our RNA-seq data set, and you can see by protein expression through flow cytometry, the long-lived effector cells really uniformly strongly express this marker. This paper showed that among the CD45 RO positive, CD8 positive subset, there were a substantial fraction of cells that also express the same marker. And this related to robust functional abilities by these cells. And for example, after CMV infection, they saw a highly enriched fraction of the cells for expressing CX3CR1. And in our mouse models, if we infect with CMV, we also see really a substantial fraction of long-lived effector cells being produced. So for this first part, I really want to convey that I think of these long-lived effector cells as first responders during a secondary infection, and that I've shown that they have rapid cytokine-driven responses, overlap with NK cells, and perhaps a unique localization. And I think they're really specialized for rapid response, both in the context of antigen exposure and through that bystander cytokine stimulation method that I described. And ongoing experiments right now are thinking about the precursor population at the effector phase that leads to the generation of long-lived effector cells, and also the in vivo contribution of the LLECs to bystander immune responses. So next I'm going to switch gears a little bit to talk about the so-called dirty mouse model. And conceptually, this idea was brought about because the realization occurred over many years and much research that the SPF mouse models we typically use in our research are probably not the best representation that we can do of a human immune system. And the hypothesis is that this responses to new infections or vaccines would be influenced by the prior immune experience of the host. And so we developed this model where the mice now have a somewhat normalized microbial exposure. And in our first description of this model in 2016, we found that the gene signatures of these mice did more closely correspond to human immune cells than did the SPF mice. So this was a team effort that initially was brought forward by Dave Massipus and Viva Aziz when they were postdocs at Emory, and they were looking at wild mice and noticing a lot of differences in the immune system compared with SPF animals. And luckily they came here to the University of Minnesota, and the discussion continued with Steve Jameson and I, and together with a really talented postdoc, Lilit Bura from Dave Massipus lab, and also some really crucial financial support from the Academic Health Center and the Center for Immunology, we endeavored to make this improved dirty mouse model. And the model itself is really fairly simple. So in most of our studies, we've taken C57 black 6 mice, so just our normal laboratory strains, and housed them with pet store mice that we just go out and buy here in Twin Cities. And after these animals live together in the same cage for 60 days, we can see that a lot of pathogens, bacterial, viral, and parasitic, are transferred from the pet store animals shown here to their co-housed partners. And we also have an additional model where we can transfer just the bedding from the cages of the pet store mice, and that will also transfer many of the same microbes. And typically the mortality is low, but we do get some deaths during this process as the animals are hit with all of these different bugs. Oh, and I should also say this is all housed in a BSL-3 containment facility, and we're very careful to separate all of this work completely from our SPF colonies to prevent any type of exposure there. So after this 60-day period, if you look in the blood of these animals, you can see that the frequency of CD4 T cells is not really changed, but the frequency of CD8s is elevated. And furthermore, for both CD4s and CD8s, the frequency of CD44 high, so memory or vector memory T cells, is enhanced. And additionally, there is an increased frequency of KLRG1 positive cells, which is indicative of our long-lived effector cell population. We also noticed after this 60-day co-housing period that a number of pro-inflammatory serum cytokines are quite elevated. So this is sort of busy, but in each one of these plots, on the left is an SPF mouse, and then it follows with co-housed, the fomite bedding exposure, and the pet store animals. And basically you can see that in all of these instances, the three groups to the right have a higher level of serum cytokine than the SPF animals. And this is, like I said, after this at least 60-day co-housing period, so this suggests that these animals are still experiencing ongoing inflammation. So what I've shown you so far is that in the case of co-housing, or what I would call diverse immune experience, there's really an elevation in the number of memory T cells, particularly in the long-lived effector cell compartment. And I didn't touch on this, but Dave Massapus spoke on this when he gave a talk several months ago. He's focused on the increase in resident memory T cell populations. And because of time, I didn't include the data, but I also have evidence to show that actually the naive T cell pool in co-housed mice seems to be unaffected from SPF. So it's really changes to the memory pool that are apparent. And I'll just bring this slide up again to say that now after co-housing, we have a lot of memory cells that can, of course, respond to their specific antigen, but also all of them have the capability of responding to cytokine stimulation in a bystander fashion. So I next asked whether these co-housed mice would be more resistant to Listeria monocytogenes in this bystander way. And in fact, we did find that co-housed mice exhibited increased resistance to a primary Listeria monocytogenes infection. And that's shown here, where again, we're looking at colony-forming units of Listeria and the spleen. And you can see that an SPF mouse at the dose we're giving will have a very high bacterial load, and few of the animals will succumb to that infection. But this is an SPF animal that's seen Listeria before. It's very protected. And we see a similar level of protection in co-housed mice by the first, in this first exposure. And this is showing pet store mice as well, which are also highly protected. And then if I come back and I deplete CD8 T cells or NK cells from these animals prior to the infection, I can see that the resistance starts to go away. So this effect here is due to contributions by CD8 T cells and NK cells in these animals. So next I wanted to examine production of interferon gamma in this situation of co-housing. And to do that, I used an interferon gamma YFP reporter mouse. So here I'm showing the first hours to days after the Listeria infection. And you can see that in the co-housed mice, there are a lot more cells detected in the blood that are YFP positive, indicating that they're making interferon gamma. And then interestingly, if I look at this 24-hour time point and divide up the YFP positive cells to see what cell types are making that interferon gamma, in SPF mice, you can see that it's really dominated by monocytes and neutrophils, which has been described in Listeria literature. However, in the co-house situation, there's really a strong shift to T cells making a lot of interferon gamma. So my initial conclusion from these experiments was that co-housed mice are more, I'm using the term immune fit, than SPF mice, at least in the case of Listeria, due to increased numbers of memory CD8 T cells that can respond quickly in this bystander fashion. Okay, but it turned out my initial conclusion was not fully correct, which I'm going to go into now. So over time, I noticed there was actually quite a range in susceptibility of the co-housed mice to the infection, and that's shown here. So the SPF mice respond very uniformly, but after co-housing, there's quite a spread. And I even would get occasionally some deaths in this group, which was quite surprising given my first few experiments. So since this time, I really thought a lot about what is different between these animals here that are very resistant and do a good job of clearing the pathogen, and these animals here, which are really no different, seemingly, from the SPF. And I thought of a couple of possibilities. One is that a specific pathogen exposure would be related to this. So for example, all the mice here were exposed to MHB, and for some reason that ruined their chances of resisting the Listeria, or vice versa down here. And just for time, I'll just tell you that I have not been able to find any association with the mix of pathogens that's transmitted during co-housing. The other thing I looked at was differences in the immune cell phenotype and number in the blood, and that's what I'll go into next. So what I noticed was that the frequency of CD44 high CD8 T cells in the blood related to the resistance of the mice to the bacteria. Now this graph is a little bit flipped from the other ones that I've shown you. So here, if there's no difference from an SPF mouse, it'll be on the dotted line. And as the resistance of the animal increases, so the fold decrease in bacterial load increases, the points will get higher on the curve. So I actually found this sort of bell-shaped situation where if there were very few CD44 high T cells, the resistance was less. As the number increased, the ability of the mice to clear the infection grew. But then, really interestingly, at this high level, it faded again. So now I kind of think about sort of 3 bins. And obviously this is a gradation, but sometimes it helps to think about things in compartments. So I think about as the immune experience of the animal grows, this moderate group, which has improved immunity, and then this very high group, which has less immunity. So why would a really large number of CD44 high T cells cause this effect? My initial impulse would be to say the more the better, that we should see a better response in these animals. So one clue came from this data where I started looking at, because of the production of interferon gamma in response to IL-12 and IL-18, I looked at the expression of those receptors on the T cells. And you can see here in SPF animals, this is showing for IL-18 receptor. Memory T cells express a little bit more. This is by median fluorescence intensity. And then in the co-house situation, that moderate group really expresses a lot of that receptor. But in the highs, it comes back down again. And I saw the same thing on NK cells, and also a similar pattern with IL-12 receptor expression. So then I wanted to go back to these 2 groups and think again about the interferon gamma production after Listeria. And that's shown here. So this is, again, using that YFP reporter mouse. And I'm comparing various time points, 0, 24, and 48 hours. Again, these really early time points before the antigen-specific response has really had time to take off. And you can see that in the moderate group, as I showed you a few slides back, there's really a strong increase in the YFP signal and therefore interferon gamma in these animals. However, in the high group, there really just doesn't seem to be a lot of interferon gamma being made. Secondly, it seems to be linked to the T cells because the percent of YFP positive cells that are CD8 positive T cells is drastically reduced in these high animals. So both the receptor expression is down, and this seems to be translating in less interferon gamma being produced. host. So now I've sort of shown you, created a third bin here, where after a high level of immune experience, or very diverse, sort of struggle with the semantics sometimes of what to call it, but high immune experience is what I've settled on so far. Now we have tons and tons of memory T cells, but we start to see that some of them decline in their ability to functionally produce cytokine. So I'm thinking of this as a type of functional almost exhaustion that sets in on these T cells that's not antigen driven. So I should say I don't see any PD-1 expression in these cells. So I'm thinking of it more as an inflammation-induced exhaustion. So my working hypothesis now is that immune experience is a good thing, but only to a certain point. So our ongoing experiments right now in this area of research are to really use transgenic T cells as a tool to more fully define the model. So in the co-housed mice, in the data I've shown you now, those memory T cells are the endogenous pool, and I don't know what antigens they're specific for or what experience they have had that leads to this point. So in this case now, I'm using P14 or OT1 transgenic T cells, creating memory in an SPF environment as we standardly do, but then seeding those cells into the co-housed environment to see how that inflammatory milieu affects their functional abilities. And so far, we've seen that for antigen-driven responses, that functionality is preserved. So in this case, if we give P14 T cells, and there are specific peptides, this GP33 peptide from LCMV, the response to that peptide in terms of interferon gamma production is similar between SPF and co-housed mice. So we can also use this same scenario to then look at the bystander response to cytokines and see if that is impaired. And the important piece is that we know the T cells aren't being exposed to antigens, so we can precisely define that as a bystander-driven response. Secondly, I'm thinking about if this loss of interferon gamma is ever a benefit to the host. So I've presented it as sort of a negative context where Listeria isn't being cleared as rapidly. But it occurs to me that if you're just full of memory T cells, maybe you wouldn't want all of them to be gushing out interferon gamma in these early hours of infection. Maybe that's too much. So thinking about survival of the host and sacrificing pathogen clearance. So we can look at some of those animals and see if the clearance is slower, for example, and also look in models like Leishmania, where in this infection model, pathology is actually worsened by prior infection. So here, memory T cells traffic to these cutaneous Leishmania sites and worsen the pathology. So in this context, dampening of the functional response might be a good thing. So those are a couple aspects that I'm thinking about. And lastly, we're starting to look at human T cells to see if they exhibit similar characteristics. And if it's possible that we can really make this simple correlation between lower and higher immune experience and the functional response of the T cells. And to do that, we're comparing US and Kenyan samples, knowing that the Kenyan people, based on their screenings, are experiencing high microbial burdens of a number of things, including malaria and parasites and things like that. Okay, and then the last few slides, I just wanted to focus on thinking about this idea of immune experience in a different context. So I've drawn this model as just an example of a way one might think of response to vaccine being affected by immune experience. So in humans, for example, you might think about a child versus an adult versus an aged person. Or you might think about an adult, as I said, from like a developing country where microbial burdens are very high. And it's been noted in these situations that responses to vaccines are typically poorer or more erratic or heterogeneous. So here I'm just thinking about, why does the response to vaccination differ among healthy individuals? And could immune experience or the previous history of that individual impact how strong the response is to vaccine? So here I'm thinking about naive responses to a new antigen. And can we sort of match that up to thinking SPF, moderate co-housed, or the pet store high co-housed scenario? And so I just have a couple of examples to show you. The first is with this Trivax immunization. So this vaccine uses just synthetic peptides that are specific for a defined peptide epitope within a pathogen, for example, that you want to vaccinate against. And this is combined with adjuvants to induce really high numbers of T-cell responses. And this is, I think, typically being thought of in terms of cancer therapy models. And so if we immunize SPF and co-housed mice and look at their responses, we can see at that early effector phase in the blood, the co-housed mice respond less well than the SPF, meaning that they're generating fewer T cells that are specific for these two peptides that I chose in this experiment. And if we get out to the memory phase, you can see there's still a decreased T cell number that's been generated. And lastly, I wanted to highlight a collaboration with Hank Balfour using an EBV vaccine modality. So Hank is going to be immunizing incoming EBV naive University of Minnesota freshmen. And so we're now performing preclinical studies in SPF and co-housed mice using this vaccine. And the formulation is this GP350 glycoprotein from EBV combined with an adjuvant. And this is a TLR4 agonist adjuvant. And the GP350 is a major envelope glycoprotein of the virus that sticks out on these spikes. So it's thought to be a really substantial target for vaccination to prevent infection of cells and development of spread. So what will the response to GP350 vaccine be in mice that have this prior immune experience? And so we performed this experiment where we intramuscularly immunized SPF or co-housed mice. We gave them 2 doses of vaccine and checked the antibody levels after each, the GP350 specific antibody levels after each dose of the vaccine. And then finally we came back with a recombinant vaccinia virus that expresses the GP350 epitope to ask whether the antibody that was produced could provide effective protection from this virus. And what we found was after the initial dose immunization, the co-housed mice did have a lower GP350 specific titer. However, after the boosting dose, there was no difference between the 2 groups, and both of them clearly increased their titer after the second boost. If we don't have the adjuvant there, we don't really see much antibody being produced. These are pretty big outliers here from what we normally see. And then I also included a human serum data point, and this is a person that's 5+ years beyond their EBV exposure. So this shows you what the titer would look like in this type of individual. So then we challenged these 3 groups here with the vaccinia virus to see how the mice would do. And what we found was, this is showing plaque-forming units of vaccinia virus in the ovaries, which is a focus of that infection. And you can see in the SPF animals, they all really uniformly cleared the virus quite rapidly. This is 3 days after infection, with about 1 million PFU. Without the adjuvant, there is not really any substantial clearance of the virus. And in the co-housed mice, most of the animals did very well, but there were some that did not clear, as we see, in the SPF situation. And in looking at these 3 time points further, I saw that it didn't really correlate with the antibody level that was in the serum. And actually, these 3 were the most highly immune experienced mice in the group. So it could be that this is not just a T-cell impacting phenomena, but it may impact the response to vaccine, both in terms of T-cells and in B-cell and antibody development. So we'll continue to pursue these preclinical studies and look at more mechanistic and delve into more detail with the cellular immune response. And then ultimately, we'll be able to compare these results with the clinical trial and see if the co-housed mice are a better representation or a better predictor of the vaccine response in humans. So kind of my overall final thoughts that I wanted to just reiterate are that long-lived effector cells, I've shown you, are distinct from the other memory populations and exhibit enhanced functional responses that aid in pathogen clearance. That increased immune experience does confer a protective advantage against bystander infections, but only to a certain tipping point. And repeated or prolonged inflammation may create what I'm calling cytokine exhausted T cells. I do think that, based on what I know right now, that the antigen-specific response of the T cells will not be affected. I'm suggesting that it's just this bystander mechanism that may be depressed after a lot of immune experience. And based on our initial studies, we might say that subunit vaccines may generate weaker or more heterogeneous responses in some of the co-housed mice, and remains to be seen, but it may more accurately inform clinical responses. And I just wanted to thank a lot of people. Steve couldn't be here today, but I wanted to acknowledge his continued support as I pursue these projects. Most of the long-lived effector cell data that I showed was produced by a postdoc named Kristen Rankema. And Christy Hensler and her group are listed on a number of these projects, and they're really quite helpful as we think about, we delve into the world of RNA-seq and everything that can tell us in these various projects. For the EBV project, Hank Balfour and Sam Dunmeyer in his lab. And the Dirty Mouse group, a lot of that data was produced in large part by Mark Pearson, who is a very experienced technician that takes care of our mice and helps with a lot of our experiments. And also, I didn't talk about malaria today, but I just wanted to say that we did have a story that just came out in the current issue of Immunity, if anyone is interested in checking that out, and maybe next time I'll be able to talk about malaria and tell you about that. Finally, I'll just say, as Chris mentioned at the beginning, the Dirty Mouse colony has now turned into a core facility, and there are a number of investigators using that colony in a lot of different models. So if anyone feels like that would be something interesting for their research, I'm more than happy to talk to you about that and help you get going in that model. And with that, I'll take any questions. Yeah.
One of the hot topics right now, of course, is immunotherapy for malignancies. And what I want to ask you is, do the long-active effector cells have any role in protecting against malignancies? And if so, are there ways of upregulating them?
Yeah. I have not done experiments like that myself. I think that would be very interesting, and they probably would be beneficial because of their high functional ability, their high granzyme B production, and also the interferon gamma. A downside to the long-lived effector cells, which I did not comment on today, is that they don't proliferate very well.
Oh.
So that's why there's tends to be more of an eye on central memory T cells, because they're the ones that really undergo robust expansion. So it's kind of like, you know, more functional or more proliferative ability a little bit. So that would be the hang up there, I would say. Okay. But the other thing I'll say is that we tend to get more long-lived effector cells with increasing rounds of antigen exposure or increased inflammation. So those would be ways to maybe push to that subset if that was deemed to be the best course. Yeah.
So one of the things that we think about, like humans, is we test not really as fancy, but T and NK cell subsets. And we calculate absolute concentration. So I'm wondering if you ever thought about, or if that's common in this type of immunology research, to calculate absolute concentration of the CD8 positive subsets, and then create reference ranges for your strains. Because I'm wondering if there are any differences if you compare the absolute amount versus the percent. Percent's obviously different. And the second question, which is kind of related, is have you seen similar phenomenon if you co-house, for example, like an FEPN or a different inbred strain in an SPF environment? Does it have the same type of responses to Black6 or any other inbred strain?
In the co-housing situation? Yeah. Okay, so the first part, yeah, I didn't show any of that today, but sometimes we do use counting beads in the blood samples that we take. We like to take blood so that we can, you know, assay from the same animal over time and get more information that way.
Because we do it two ways. Like usually we take the white count based on automated hematology analyzer and multiply it times %, or we can use beads and kind of what we call a single platform. What's nice is that if you, I think it's analogous to dogs. So if you look at, dogs are lots of examples of inbred species, right? You have inbred dog breeds.
Yeah, sure.
The reference ranges is different. Like a dachshund is different from a German Shepherd.
Yeah.
I think you could gain a lot of information by looking at your different house groups by creating reference ranges for each subtype.
Yep, that's a very good point. So the vast majority of our work has been in the B6 model. And that's kind of driven by the fact that so many knockouts and reporter strains are available on that background. So that tends to be what we focus on. We have done some work with BALB/C mice. Initially, we found a lot higher mortality with BALB/C in co-housing. And so we kind of got scared off of that and went back to B6 for a while. But now more people have been going back to both the co-housing and also using the bedding, the fomite bedding transfer, because we see very low, if any, mortality when we just transfer the bedding. And one issue that has come up is kind of just what you're suggesting, that we don't see as strongly the same signature changes in the blood in BALB/C that we do in B6. So the BALB/C mice tend to already have a higher CD44 component, for example. So we're actually starting some new experiments to see, just to get more data on the blood analysis and compare to SPF. Maybe there's some other marker that will emerge as better in BALB/C than B6. And also I want to follow that up with the Listeria infection experiments to see if the markers that we highlight also translate to the step of improved protection. So we have a lot of work to do with various strains, but I agree with you, that would be really nice to get to that point.
exposure model in some of the newer mouse models of disease that, represent the human immune system.
Yes, I do.
Either like an Alzheimer's disease or TREM2, something like that. I think that would be really awesome. Yeah.
Okay. Yeah, that's an interesting idea. I'll tell you what we are planning on doing. I'm not exactly sure if it's where you're going, but for the EBV model, because EBV does not infect mice naturally, I'm now going to start trying to develop a humanized mouse model, which has been used in EBV research. Not a dirty humanized mouse model, so that'll be the extra step that I'll try and take and see if we can build on that. But I think that would be really cool, and that would even get us closer to models that may be really helpful in thinking about human disease.
And then my second question was just your list of all of the exposures.
Yeah.
Just all that were there, or just?
No, that is like the standard RAR panel. That they screen. So if you notice, like the laboratory strains, those are 0 for all of those pathogens. We have done a little bit delving deeper into the parasites, the microbiome, those sorts of things, but it gets very costly to do on every single animal. So the simple serology panel is definitely the one we've used most commonly because of that.
Very nice talk, Sarah. So I'm curious about what you think or the field thinks about this overlap of the RNA-seq data from the long-lived effector cells and NK cells. What is Mother Nature or somebody telling us?
Yeah. Well, I think it's pretty cool.
Yeah, I know. But what does it mean, or how did it happen? Or,
Yeah. I guess I think that the memory pool is really, there's really much more overlap than we've even
it's like a redundancy
appreciated to this point. Yeah. So probably overlap within the T-cell pool and also with other immune cell populations. And yeah, I just think it makes me think that the long-lived effector cells are really focused on providing this very early control to like keep things in check until the antigen-specific response can kick off, and probably the central memory T cells will then rapidly proliferate and contribute to the overall immune response. So that's kind of what it says to me, is just points to the rapid nature of their ability to respond. Yeah,
Yeah, I was wondering for the moderate versus the high immune experience animals, if you think that that is a difference between kind of more acute exposure and the inflammation that's going on with those acute infections, or whether you think that effect will persist over time. Because I think in humans it's fairly well accepted that sort of the acute infection can depress your immune system, especially in certain situations, influenza or other things. And I'm just wondering if you've looked at the time course of that at all.
Yeah, I think that's super interesting. So I'll say that all of these experiments take place at least 60 days after the animals have been, put together with the pet mice. A lot of times, unfortunately, it's a lot later than that. The mice are sitting around. However, that doesn't mean that all the exposure occurred on day 1, right? The animals could reactivate for some virus later and things like that. I will say when we bleed over time, we don't see a lot of spikes in like the frequency of T cells or the CD44 high nature. It kind of goes up and then it stays, or it kind of declines a little bit over time. So as much as I know from that, I don't think they're undergoing acute infection, but I have thought about trying to mimic the situation by either intentionally infecting the mice with a much more robust mixture of pathogens than we would normally use, and see if the same kind of themes emerge, and I can kind of reductionist way recreate it. So then I can know, these are acute pathogens, they're gone, that type of approach. But I think that's really interesting to know whether it'll really be there long term, or can it be corrected later on.
Yeah, I had actually a similar question, because in humans, as you probably know, we check the temperature and make sure the patient doesn't have acute infection at the time of vaccination, because then it wouldn't work. So can you take temperature in mice? Can
you
can.
can.
Level? Can you do something like that?
I've thought about doing that. You can do that. So that might be something.
The next question I had was, where are the long-lived effector memory cells determined? Are they already present in the thymus, or is it something that happens later?
Later. So they're memory cells after the initial response by innate T cells to antigen, and then they develop after that point.
Because we have recently paid attention to CD161, which is the human counterpart of NK1.1.
Okay.
And we found that this is a fairly good marker of probably about half of T cell lymphoma of a certain type. And I was wondering, what does it correspond to? So maybe it corresponds to, I was thinking maybe it's a stage of timing development, but alternatively.
Do you think they could be recirculating back from the periphery through the thymus, or? It's
presenting as leukemia, so they're everywhere at the time of presentation. Okay. It's just we know that lymphomas sometimes sort of reproduce phenotype of certain cell type existing in nature. And there are two steps that I'm aware of in humans. One is called NAIT.
Okay. Yes, innate cells. Yep.
Are they in any way related to your population?
I don't think so. So these are CD8, alpha beta kind of standard T cells that I've described.
The NAITs are defined by certain alpha beta.
Restricted, right?
Yeah, I don't know if you check that. How I know that?
I have not personally checked that, but I don't have reason to believe that there's a restricted T cell receptor.
And then there are papers that all the T cells expressing CD161, they have this IL-12, IL-18
high expression,
which they claim is. So I think there is some overlap.
Okay, thanks. Yeah, I'll look into that. Think about it. Yeah.
I like your talk, Sarah. There's a lot of food for thought here. What I have trouble dealing with is your slide that relates to antigens and vaccine and responses. So if you gave a certain antigen to all of us in this room, immunized us, you'd have a great diversity of immune responses, probably regulated by the immunogenetics of all of us. So the mouse breeds also reflect this type of immune diversity, but it's very tricky because you can manipulate the antigen biochemically in different ways. You can mutate it in order to enhance or mute the immune response. So I think it's really tricky interpreting some of the data at times.
Yeah, I agree. It's making a big leap to say that explains why people respond differently to vaccines. I don't want to go that far with it, but I think that this could be one factor that is potentially influencing the situation. But yeah, it'll be really hard to tease apart the genetic component from inflammation. Yeah.
Great thought. Thank you. And put it all into English, which was even better. CD44 high, we're interested in CD44 for a number of reasons. And this seems to be characteristic of memory T cells, at least in mice, is that fair?
Yeah.
And so the question is, it's an adhesion molecule.
Yeah.
Binds to T proteins, binds to a lot of things, helps growth factors, stimulate responses better, and so on. So it does a lot of things. So have people looked at the effector response in CD44 null mice to see whether there's an impact either in the frequency of these cells or, more importantly, their tissue distribution in terms of where they go?
I believe there's no impact.
There's no impact?
Yeah, I believe that's true. So it's one of those situations which unfortunately is sort of common in the memory T cell field, where a lot of these markers, they're nice markers, right, but the functional reason that they're there is maybe not apparent to us so far. Yeah.
So it has been done. Those experiments have been done.
Yeah.
Okay,
Yeah.
thank you.
Okay, thanks.
Thank you.